Hello and welcome. Let's continue today with digestive tract and digestive system. So here when we go through and we talk digestive tract, digestive system, here we've got, now we can see, we'll be able to appreciate, going back actually first, we went through and we can see here, we talked about stercobilin and how stercobilin was going to be a breakdown product of heme, and it was going to be giving our feces its brown color. Now we have the same thing here with urine. Now in relation to urine, we're going to have what we call urobilin or urochrome. So in relation to urine, just like we have with feces, stercobilin, for urine we have urobilin or urochrome, and that will be basically the pigment found in urine, giving urine its characteristic kind of color there as well. So here now, when we go through and we talk about bile, we ended the last section by talking about how in the absence of bile, feces are going to be gray, white, and they have fat in them as well, and that's because no fats are digested or absorbed. So bile is important there as well. Now, if we talk about having the gallbladder removed, now if we have a patient, we tell our patient who's had their gallbladder removed, what are we going to tell this patient? Are we going to tell this patient not to consume any fats or to decrease the quantity of fats? Now there we're going to tell them to decrease the quantity because the gallbladder being removed, that's the storage form. So bile is still going to be produced because we said it's going to be produced by the liver. And we'll talk more about that when we talk about it being released there as well. So bile is very important. Let's talk then pancreatic juice, and let's talk pancreas. Now before we get into pancreatic juice, let's talk pancreas. Here we can appreciate the pancreas. The pancreas is going to be found located right near that duodenum of the small intestine. You can see just inferior to that stomach, and it spreads basically across that abdomen, and you can see it's going from this duodenum, and here you have the head of that pancreas. Here you have the tail. The tail is going to be basically tickling the spleen because the spleen is going to be found, you can see right adjacent to it. Now here the spleen, now you can see, is going to be found in basically the left side of that abdomen, we could say. Now here we've got then the pancreas. Now the pancreas itself has got two ducts. You can see here we've got first the main pancreatic duct, and then we're going to have the accessory duct, which is going to be the smaller duct that empties into the duodenum proximal to the main pancreatic duct. And then the main pancreatic duct is joined by, if you recall, the bile duct, then here, which is coming from the liver, and then also the gallbladder, the cystic duct, and then basically within the liver you had a whole bunch of smaller bile ducts that came together to give rise to the common bile duct, and then that duct joins the cystic duct to become basically this bile duct that we see here. Now main pancreatic duct coming in and the two coming in joining together, and here's where you could see they'll allow both of their secretions to make their way in. So let's talk pancreas then. Now when we talk pancreas, the pancreas has got dual function, endocrine and exocrine. We've already talked exocrine, we've already talked endocrine, right, when we did endocrine system. Now exocrine is going to deal with digestive tract and digestion. So here when we talk now, pancreatic juice is the main component. You can see here we produce about 1,200 to 1,500 milliliters a day. It's a watery alkaline solution that's going to neutralize chyme. It has a pH of about 8, right? If you recall chyme is going to have a very acidic pH because that chyme is coming from the stomach. In the stomach, we saw that pH there to be very acidic sometimes. Now, electrolytes we're going to see primarily bicarbonate is going to be found within pancreatic juice. And again, helping to create that alkaline solution. There's enzymes that are going to be found within this pancreatic juice as well. These enzymes are going to include amylase, lipase, You could see nucleases, which are going to be secreted in active form, but they require ions or bile for optimal activity, and then proteases, which are going to be secreted in inactive form. So when we go through and we talk about pancreatic juice, it consists mainly of water, and it contains enzymes and electrolytes. Electrolyte primarily bicarbonate ions. And the pancreas secretes enough bicarbonate and balances the amount of hydrochloric acid that's produced by the stomach. So this is why we have this alkaline solution. Like pepsin of the stomach, pancreatic proteases, okay, pancreatic protein digesting enzymes are produced and released, we said, in the inactive form, which then get activated in the duodenum, where they will actually do their work. This now prevents that pancreas from digesting itself. In conditions like pancreatitis, the enzymes are activated as soon as they are produced, so it basically causes that pancreas to digest itself. Now, for example, within the duodenum, we have enteropeptidase, which is an intestinal brush border protease. It activates trypsinogen to trypsin. You can see that there very well. And also then chymotrypsinogen to chymotrypsin and procarboxypeptidase to carboxypeptidase. Other pancreatic enzymes I mentioned are going to be proteases. Now, when we talk proteases, they're there to digest proteins. We have lipases. We mentioned there as well. Fat splitting enzymes. Nucleases, enzymes that are going to be breaking down DNA and RNA nucleotides. These nucleic acids to nucleotides, basically. And here, when we go through and we talk about nucleases, lipases, and these proteases, as we said, they're going to require either ions or bile for optimal activity. Now, when we go through and we talk about protease activation in the duodenum, you can see trypsinogen we saw is going to be activated to trypsin. And then we saw the same thing with procarboxypeptidase and chymotrypsinogen. Next, then we move down into the regulation of bile and pancreatic secretion and entry then into the small intestine. Now, secretions, we'll see bile secretion. Okay, bile secretion is stimulated by bile salts. We're going to go through, we're going to check that out, we're going to see. And that's going to be bile salts that are going to be found in the enterohepatic circulation. We could see bile secretion is going to be stimulated by bile salts and secretin. Now, when we go through and we talk about this regulation, you'll see secretions of bile and pancreatic juice and its release are regulated by, first, let me tell you, neural stimuli and also by hormonal stimuli. Neural and hormonal stimuli. Hormones are going to be CCK, cholecystokinin, and secretin. We could see right there, as we mentioned. Bile salts themselves are a major stimulus for bile also, as we mentioned. Now, when no digestion is occurring, you'll see here, bile is going to start to fill into the gallbladder. It kind of starts to back up. When it starts to back up, here, let's see, did we have a good picture of this? Okay, right in here. So here we could see, if we were to draw in, let's draw in here. So here you have basically the cystic duct, and then here you can see you've got now this common hepatic duct. They come together and they become the bile duct. So here the bile duct is emptying into here. So now when this hepatopancreatic ampulla is closed, there's no digestion occurring. Now this is going to be closed. Now bile is going to start to accumulate, and it starts to fill right up into here. Starts to fill up into that cystic duct and allows it to then store up into the gallbladder. And the gallbladder, what it does is it's going to start to concentrate it. And now here then, basically now if that fills up, then obviously more is going to continue, and it'll then basically fill right up into the liver, then allowing the liver to know, hey, we've got more than enough produced here. So here now, then moving back away, we could see when we go through and we talk then, so CCK and secretin, and then bile salts themselves we've mentioned there as well. So when there's no digestion occurring, the bile is going to fill into the gallbladder. Bile is going to fill into the gallbladder. Okay. So if that hepatopancreatic sphincter is closed, okay, unless digestion is active, then it's going to be open. Bile is going to get stored into the gallbladder, as we can see here. So we said bile fills into the gallbladder and isn't released until the major stimulus, CCK, which is an intestinal hormone, is secreted, which causes the gallbladder to contract, and when the gallbladder contracts, that's going to cause bile to be released. Now here we could see CCK, okay, neural stimulation. We've got here, we could see vagal stimulation, and it's a minor stimulus. And then CCK is also going to cause the secretion of pancreatic juice and the hepatopancreatic sphincter to relax. So you got to think of these things all are going to be happening at basically simultaneously now when digestion is taking place. CCK is going to induce secretion of enzyme-rich pancreatic juice by acini as well. And here we can see secretins functions, causes secretion of bicarbonate-rich pancreatic juice by duct cells. And then vagal stimulation also causes release of pancreatic juice, and again, that's a minor stimulus. Here we could actually see now how all of these processes are going to be intertwined now. Here we can see chyme is going to enter. Chyme entering into the duodenum causes now the duodenal enteroendocrine cells to release CCK and secretin. So here we could see in red and yellow dots, CCK in red dots and secretin in these yellow dots, they will enter the bloodstream. CCK induces secretion of enzyme-rich pancreatic juice, as we said. Secretin causes secretion of bicarbonate-rich pancreatic juice. And then bile salts. Bile salts, and to a lesser extent, secretin, which is going to be transported via the bloodstream, are going to stimulate the liver to produce bile more rapidly. CCK, we said, makes its way then via the bloodstream, making its way to the gallbladder, causing then the gallbladder to contract, and we said the hepatopancreatic sphincter to relax, allowing bile then to enter into that duodenum. During then the cephalic and gastric phases, vagal nerve stimulus is going to stimulate the gallbladder to contract then, but in a weak fashion. So then when we look at now the digestive processes that are going to be taking place within the small intestine, We'll see first, I want you to know when we talk about the small intestine is the body's major digestive organ. Digestion is complete here in the small intestine. Virtually all absorption occurs here as well. Absorption of most of our water and all nutrients is going to take place here inside the small intestine. Like the breakdown products of carbohydrates, proteins, fats, nucleic acids, vitamins, electrolytes, and water, basically, all is going to take place here in the small intestine. The small intestine has no part in ingestion or defecation. Now, when we go through, we talk about the digestive processes that are going to be taking place. You can see here, chyme from the stomach is going to make its way then into the small intestine, and chyme itself contains these partially digested components here of carbs and proteins and undigested fats. Lingual lipase contributed a very small amount. Here now, we'll see when bile is able to act, and then our pancreatic enzymes are able to act, we're going to see digestion of fats take place. So the bile is very important there as well. Now, when we talk digestion in the stomach, you'll see digestion in the stomach 3 to 6 hours it can take. And so when we talk, that'll all depend on again, the contents of what you've consumed, high fat, high protein, or high carbohydrate. So each of these will take again their own considerable amount of times. Now all nutrients are going to be basically absorbed here. We said as well. Now small intestine, like the stomach, have no role in ingestion or defecation. Now, the digestive processes that are going to be occurring in the large intestine, we'll see no food breakdown occurs in the large intestine. What gets delivered to the large intestine contains few nutrients. The large intestine is going to harvest vitamins, K and B complex vitamins, and they're going to be made by bacterial flora. We'll check that out here in a few seconds. And this large intestine is going to absorb any remaining water and electrolytes. The primary task of the large intestine is to propulsively force fecal material towards the anus and eliminate it from the body, basically during defecation. Now we talk large intestine. The large intestine, we'll see then, is going to get divided up into a few different parts. Now when we go through and we talk about the ascending, transverse, and descending colons, you can see here when we talk colon, the colon is not essential for life. Now here what can happen is if a patient has any type of a problem, if the colon has to be removed, the terminal ileum then is going to be brought to the abdominal wall. It gets brought to the abdominal wall in a procedure that's called an ileostomy. And food then gets eliminated into a sac. The ileum can also be attached to the anal canal as well. Now, when we go through and we talk the digestive processes that are taking place in the large intestine, you'll see here now this large intestine, its tasks can be anywhere from 12 to 24 hours. Again, no food breakdown except by enteric bacteria. Vitamins, right, that are going to be made by bacterial flora. We said K and B complex vitamins. Water and electrolytes are going to be reclaimed. So just a few processes that are taking place here. Vitamin formation, which vitamins? We mentioned there. And then water and electrolytes are going to be reclaimed. So the major function is a propulsive function, which is to force basically feces towards that anus, we said, for defecation then. So here we can appreciate then a nice table depicting to us what's happening inside the small and large intestines. Now here as we progress then, here we could look at then the physiology of digestion and absorption. Now let's go here then and see the entire chemical processing, the enzymatic breakdown and absorption of each class of foodstuff as it moves through the GI tract. So let's get into then basically digestion. Let's talk chemical digestion, more specifically speaking, right? Versus mechanical, which we saw was because of teeth, tongue, and an extra layer on the stomach there as well. So when we go through, we talk digestion now. Chemical digestion. Chemical digestion is going to involve, we'll see enzymatic hydrolysis. It will involve enzymatic hydrolysis. So when we talk enzymatic hydrolysis, we're talking the use of enzymes, and we're talking hydrolysis, we're talking the use of water here as well. So enzymatic hydrolysis, like we said, will be involving enzymes plus hydrolysis, which is the addition of water there. Now, when we go through and we talk digestion, digestion you'll see is going to be a catabolic process. It's a catabolic process in which large food molecules are going to be broken down to monomers, which are the chemical building blocks, which are going to be then small enough to be absorbed into then the GI lining, and then through the GI lining, basically we'll see it'll make its way out into then the blood, basically when we get into absorption and we talk about there then. Now, when we go through and we talk then digestion is going to be accomplished by enzymes. It'll be accomplished by enzymes. Now, the enzymatic breakdown of any type of food molecule is called hydrolysis because it involves the addition of water molecules to each molecular bond that is to be broken or that's to be lysed. Excuse me. Now, when we go through and we talk digestion, first we'll go through and we'll talk, so again, catabolic steps, where our macromolecules will be broken down into monomers, which are going to be small enough for absorption. Now, enzymes are going to be involved, and we're going to see intrinsic and accessory gland enzymes are going to break down foods, and then the hydrolysis addition of water, which is going to be involved there as well. Now, when we go through, first we'll look at the digestion of carbohydrates. Now, when we talk digestion of carbohydrates, only monosaccharides can be absorbed. Monosaccharides, simple sugars, okay, which are the monomers of carbohydrates, they are absorbed immediately. They're the only, basically, monosaccharide, when we talk, are going to be the only component that can be absorbed when we talk about carbohydrates. We talk disaccharides, polysaccharides. We'll see they're not going to be able to be absorbed. They have to be broken down to a monosaccharide then to be absorbed there. There's only three of these three common monosaccharides in our diet. They will include glucose, we can see here, fructose, and galactose. So glucose, fructose, and galactose. The more complex carbohydrates that our digestive system is able to break down to monosaccharides are going to be the disaccharides, which include sucrose, if you recall from the beginning of the semester, table sugar. And we saw that was formed from glucose and fructose, so make sure you review how they're formed. Lactose, milk sugar, and then maltose, which we described as grain sugar. And also the polysaccharide, glycogen, and the polysaccharide starch. You'll see, the intestines can absorb only monosaccharides. Therefore, all dietary carbs must be digested to monosaccharides to be absorbed. Humans lack enzymes that are capable of breaking down polysaccharides, such as cellulose and chitin. Cellulose, if you recall, is going to give our plant cell walls structure, basically strength. And then we talk chitin. Chitin is going to be a fibrous substance in the exoskeleton of arthropods, and also it's found in the cell walls of fungi as well. Now, when we go through and we talk the average diet is going to, you'll see most average diets are going to contain 60% digestible carbohydrates. And these carbohydrates are going to be found in the form of starch. Starch, disaccharides, or monosaccharides, mostly starches. Now, we talk digestion of starch. Digestion of starch begins in the mouth, and we said it begins in the mouth thanks to salivary amylase. And it's going to continue, it'll continue until these contents reach the stomach, where then amylase becomes inactivated. Starchy foods or other digestible carbohydrates that don't get broken down by salivary amylase, they then are acted on by pancreatic amylase, which is going to be found coming into the small intestine. Intestinal brush border enzymes will further digest these products to monosaccharides. Some important brush border enzymes are going to include, we'll see, dextranase, glucoamylase, which act on, we'll see, oligosaccharides, and then maltase, sucrase, and lactase, which hydrolyze maltose, sucrose, and lactose, right, these disaccharides, down to monosaccharides. And then when we talk salivary amylase, you can see it's a pH basically inside the mouth versus what you have in the stomach, so it becomes inactivated. And then we have pancreatic amylase, which is going to be coming into the small intestine, which is going to continue to break down basically then whatever oligosaccharides or any other carbohydrate that may have been not broken down thanks to salivary amylase. And then we have our brush border enzymes we have mentioned. So here, then we're going to see are those basically monosaccharides that are ready to then be absorbed. So we'll look at them then again when we get to absorption. So let's move then to, here it goes through and tells you basically the same thing that we went through, we just talked about. And the absorption is in here as well. We haven't talked about that. We'll talk a little bit about that too. Now, we talk digestion of proteins. Protein digestion begins in the stomach when pepsinogen, which is secreted by chief cells, gets activated to pepsin. When pepsinogen secreted by our chief cells is activated to pepsin. Pepsin's function, you'll see, is going to be then to break down these proteins. Protein denaturation, we'll go through and we'll see. Now, when we go through and we talk about pepsin, pepsin functions optimally in the acidic pH that's found inside the stomach. It functions optimally in this acidic pH. Okay, here we can see when we go. So as we're saying, pepsin functions optimally in that stomach because of that pH inside of that stomach. The pH inside the stomach is about 1.5 to about 2.5 there as well. Now here when we go through and we talk this pH, here you could see, so it can vary anywhere from, what we have in there, depending again on stomach contents as well. Now, here when we go through and we talk about pepsin though, pepsin is going to be inactivated now by the high pH of the duodenum. Now, it gets inactivated as soon as it makes its way inside the duodenum. Protein entering the small intestine then is going to be acted on by trypsin, chymotrypsin, you could see there, and then also carboxypeptidases are going to kick in as well. So again, protein entering the small intestine is going to be acted on by trypsin, chymotrypsin secreted by the pancreas, okay, which cleaves the proteins into smaller peptides. And then carboxypeptidases, aminopeptidases, and dipeptidases, which are going to be pancreatic and brush border enzymes, are going to liberate the final amino acid products by dismantling the proteins. And then here we could see basically all of those final products now right inside of here. And then we're going to talk about this process again, absorption, in a few minutes. So here you can see you had all those amino acids and basically protein fragments. And here you can see all those simplified amino acids or liberated amino acids. Next one, when we talk lipids, then here we're going to be able to see, now when we talk lipids, digestion of lipids, first, triglycerides are going to be the most abundant fat in our diet. And the small intestine is going to be the primary site of lipid digestion because the pancreas is going to be the only significant source of fat digesting enzymes, or lipases, we could say. Now, since we know triglycerides and their breakdown products are insoluble in water, fats are going to need a special pre-treatment. They require a special pre-treatment or a process known as emulsification, and that's going to involve bile salts. So again, when we talk about fats are going to need a special pre-treatment with bile salts to be digested and absorbed then in the small intestine. Now, bile salts, what they do is they're going to help break off little fat droplets from large fat globules and form an aqueous suspension of fatty droplets that are going to be called an emulsion. Emulsification, then, is a process that these particles go through to be more widely dispersed, or where they're going to have their surface area increased. Why do we want to increase the surface area? If we increase the surface area, we can see then when enzymes act, they have more optimal digestion basically take place, and they basically have more surface area to act on, leading to then again more optimal digestion. Now, this process, again, it increases the number of triglyceride molecules exposed to the pancreatic lipase. That's the whole thing there. Without bile, lipids would be incompletely digested. Pancreatic lipase is going to catalyze the breakdown of fats. Digestion of fats by pancreatic enzyme lipase yields free fatty acids and monoglycerides. We can see that. These then associate again. Okay? They're going to associate again, and they're going to associate again with bile salts to form what we refer to as micelles. They're going to form then micelles, and they'll be able to then diffuse right into the intestinal basically cells and right through the intestinal mucosa, you could say. Fatty acids and monoglycerides are going to leave micelles, and they're going to diffuse into the epithelial cells. So again, now when we talk micelles, when we go through, and you can see here, digestion of fats by the pancreatic enzyme lipase is going to yield free fatty acids and monoglycerides. These then associate again with bile salts. They associate then again with bile salts. Okay, so here when they associate it again with bile salts, they form micelles. These micelles we see here are going to ferry these fatty acids, and monoglycerides. They're going to ferry these fatty acids and monoglycerides basically to these cells. Now they bring them right to that intestinal mucosa. Then what happens is the fatty acids and the monoglycerides are going to leave the micelles and diffuse into the epithelial cells. And then we'll talk about absorption. Here you can see eventually they're going to make their way either into the lacteals or into the bloodstream. So when we get into absorption, we'll look at that right down here, then in a few minutes. Let's talk about then digestion of nucleic acids. When we talk about the digestion of nucleic acids, pancreatic nucleases. Pancreatic nucleases break down nucleic acids to basically nucleotide monomers. And then intestinal brush border enzymes, nucleosidases, and phosphatases, are going to break them apart to release a free base, basically, pentose, the free pentose sugar, then to free the pentose sugar, and also to free that phosphate ion. So when these nucleosidases and phosphatases act on the nucleic acids, they're going to help liberate the bases, the sugars, and the phosphate components that we saw making up these deoxyribonucleotides and ribonucleotide nucleic acids in the beginning of the semester. So we looked at carbohydrate digestion, protein digestion, fat digestion, and then nucleic acid digestion there as well. Let's talk about absorption then. When we talk absorption, mostly all of our foodstuffs, okay, 80% of the electrolytes and most of the water are reabsorbed in the small intestine. Remember, water follows salt, as we have talked about in relation to the kidneys. Same thing is going to be happening here. Now, at the end of the ileum, all that remains is some water, indigestible food materials like cellulose, and also millions of bacteria, which gets passed on into then the large intestine. Most nutrients are absorbed through the mucosa of the intestinal villi by active transport driven by ATP. Thanks to ATP. They then enter the capillary blood. They will then enter the capillary blood in the villus, basically to be transported then eventually into the liver. Right? That's the gatekeeper there. So let's talk then carbohydrate absorption. The monosaccharides glucose and galactose, liberated by the breakdown of starch, are going to be shuttled by secondary active transport with sodium into epithelial cells. With sodium into the epithelial cells. They then move out of these cells by facilitated diffusion. They will then move out of these cells by facilitated diffusion. And then they're going to pass into the capillaries, right? Next in here, when we go through, when we talk about fructose, you can see, moves entirely by facilitated diffusion. It moves entirely, so it enters and it exits the cells entirely by facilitated diffusion. So here we could see that. Glucose and galactose were liberated from basically disaccharides and polysaccharides, and these are the monosaccharides that were created there. So here you can see they're going to be absorbed, as we described, by secondary active transport with sodium. Secondary active transport with sodium. So sodium comes in, and that's how glucose and galactose then are going to be able to make their way in. And you can see then that glucose and galactose, whichever one is making its way in, plus sodium then are inside the cell. And then the sodium, you can see, is going to be basically moved out via the sodium-potassium pump. And then glucose and galactose, we said, are going to now pass out of this cell, right? So we said then they move out of these cells by facilitated diffusion and pass into the capillaries. And here the receptor is known as the glucose transporter 2, GLUT2 transporter. So you got to make sure you have that down too. Next then, let's talk protein absorption. Now we talk protein absorption, then several types of carriers transport the different amino acids resulting from protein digestion. Okay, so amino acids are transported by several types of carriers. And most of these carriers are coupled to the active transport of sodium. Most of them are going to be coupled to the active transport of sodium. Other carriers use other active ways, like hydrogen ion dependent cotransport as well. Okay. Then we'll see the amino acids are going to leave the cell and pass into the capillaries by facilitated diffusion. Thanks to facilitated diffusion. So here we could go moving back here, we could see sodium, hydrogen ion as well sometimes. So whatever the case may be there, those amino acids make their way in. Once they're inside these cells, then they're able to make their way out of the cells, as we said. And when they make their way out of the cell, that's going to be, again, we said, facilitated diffusion. And they'll make their way then into the capillaries. Now, capillaries, we're saying here, capillaries we saw with carbohydrates. Which capillary is this? Which blood vessel is this? Eventually making its way to, right? Small intestine, if you're talking there, right? Large intestine, stomach, all these different areas. But mostly this absorption, we said, is going to be taking place in the small intestine. And if you recall from anatomy there, you've got the hepatic portal circulation. Now, when you talk hepatic portal circulation, you've got there superior mesenteric vein, inferior mesenteric vein, right? And superior mesenteric vein, splenic vein, inferior mesenteric vein, they all come together to give rise to the hepatic portal vein, which will then allow for these basically components to make their way then into the liver. Excuse me. So here then we can see now when we go through and we talk about lipid absorption then. Let's talk lipid absorption. Now we talk lipid absorption. Lipid absorption now, here we could see upon reaching, okay, so upon reaching the epithelial cells, the lipid substances are going to diffuse through the lipid membrane, and they're going to diffuse through the lipid membrane by simple diffusion. Once the free fatty acids and monoglycerides enter the epithelial cells, you'll see the smooth endoplasmic reticulum converts them back to triglycerides. Now, when we go through, we talk then, here you can actually go through and you can look at this whole process as we went through. You can read through basically this whole process right inside of here. They're going to be combined then with lecithin, phospholipids, and basically cholesterol to form chylomicrons. Okay, so chylomicrons are going to be important, and then chylomicrons are going to be removed by exocytosis. The chylomicrons are going to enter the lacteals, and then they get carried away from the intestine in the lymph. So here you can see that when you move back to this process right inside of here. And then here is where we were talking then. So chylomicrons, going to be basically with long chain fatty acids, right? They're going to be passed through the lymph. And then we talk absorption of glycerol and short and medium chain fatty acids. That'll be basically, they'll be making their way right into the capillary blood in the villi. And then again, hepatic portal vein, hepatic portal circulation, as we saw there. Vitamin absorption. Let's talk vitamin absorption. Absorption of nucleic acids, actually, before we jump down. So we talk nucleic acids. Nucleic acids, we talk nucleic acids here, then you could see transport, active transport across epithelium and into the bloodstream then. So vitamins, small intestine absorbs dietary vitamins, and the large intestine absorbs some of the K and B vitamins that are going to be made by its bacteria. Vitamin B12, the vitamin B12 and intrinsic factor are absorbed at the terminal ileum, as we've described, at the terminal ileum. So small intestine, fat-soluble vitamins, again fat-soluble, so they're carried by micelles, and then they diffuse into absorptive cells. Water-soluble vitamins, they're going to be absorbed by diffusion and or by active or passive transport. And then B12 binds with intrinsic factor, and that's going to be absorbed by endocytosis at the terminal ileum. And then large intestine we talked about there. Absorption of water. 95% of water is going to be absorbed in the small intestine, while the rest then it gets absorbed in the large intestine. And the small intestine then, it'll happen thanks to osmosis. And then water uptake is coupled with solute uptake also. Concludes digestive system.